Hi, engineers. In this video, we're going to talk about the motility of the small intestine. So let's go ahead and get started. All right, guys. So when we talk about the small intestine, we know a lot of its functions are pretty primarily absorption. It plays a significant role in absorption. Okay, we talked about that a lot when we talked about the videos where we discuss carbohydrate absorption. We talk about protein absorption. We talk about lipid absorption. So very significant role with that. We also talked about how it regulates the gastric secretion and gastric emptying processes. We talked about that in the gastric motility. We talk about it in the cephalic, the intestinal, and the gastric phases of gastric secretion. So we know that it performs a lot of those different functions. Motility is another important one. Motility allows for it to be able to allow for mixing movements, helps with absorption, and also move it towards from the oral side to the anal side. So the first one that we're going to talk about, the first motility that the small intestine does, is called segmentation. So let's write that down here. So big thing with segmentation, okay, is it plays a significant role in mixing. So here's the couple of things I want you guys to remember about segmentation. It's very sluggish and, let's say, short-lived contractions. Okay, so they're very sluggish, very short-lived contractions. They're not super powerful contractions. These are specific primarily for mixing. And what do I mean mixing? So whenever food is in the small intestine, right, it's considered to be chyme at this point because it came from the stomach straight into the duodenum, the first part of the small intestine. What we're going to do is we're mixing chyme with a bunch of the fluid that's in the small intestine. Well, what's in the small intestine? Well, if you guys remember, there's bile. And where'd that bile come from? It came from the liver and it came from the gallbladder down that biliary tree. So we're going to try to mix the chyme with the bile. Why is that important? What does the bile help for? You guys remember, it helps with emulsification of fats to allow for better digestion or chemical digestion specifically of fats so that we can absorb fats, cholesterol, and fat-soluble vitamins. So that's a very important one. We also want to mix the chyme with the pancreatic secretions. So remember the pancreatic juice? So the pancreatic enzymes are one component of that. What are some of those enzymes? You guys remember there's pancreatic proteases, pancreatic lipases, pancreatic amylases. They help with breaking down the protein in the chyme, the carbohydrates in the chyme, the lipids in the chyme. So it's a significant thing. And we want to mix it with the intestinal fluid. So we have a lot of intestinal fluid, which is rich in bicarbonate and mucus. Okay? So we have intestinal. And what other fluid? What comes with the pancreatic juice? Pancreatic enzymes and bicarbonate. So intestinal and pancreatic fluid. And the big component of this is I want you to remember that it's rich in bicarbonate and mucus. Okay, so that's the big thing I want you to remember with segmentation. What's the primary role of segmentation? If you forget all of these things I'm going to talk about, please don't forget that it mixes chyme with a bunch of secretions. Why? Because it enhances absorption. So the whole goal of mixing the chyme is to enhance absorption. What's something else that it does? So in segmentation, it's sluggish, short-lived contractions, right? And we're going to explain the mechanism in a little bit, but it moves things anterograde, which means it's moving it forward, right? So towards the anal side. And it also performs retrograde movement, which means it's moving towards the oral side. And that is what allows for this mixing. So it's called a to and fro movement. It's contracting and it moves things forward and it moves the chyme backwards. And that whole process allows for mixing. What's the other thing it helps for? It increases the contact time with chyme and mucosal surface. And again, this goes back to the overall goal, which is to do what? Stimulate or enhance absorption. That's the goal. So we want to mix the chyme with the actual bile, pancreatic enzymes, intestinal pancreatic fluid to enhance the absorption and chemical digestion. But we also want this segmentation so that way whenever this is contracting, it's causing an increased contact between this chyme bolus with the mucosal surface. Why the mucosal surface? Because that's where absorption is occurring. Okay? So what do we need to know about segmentation? Okay? All right, so look how this works, right? We have different points of the small intestine where it's basically, whenever the food bolus comes into the small intestine, it causes distension. And that distension is what triggers a local reflex arc, right? If you guys don't know what I'm talking about, watch our video on the enteric nervous system. We go over the short reflex arcs and the long reflex arcs. For right now, I just want you to trust me that whenever food is in the small intestine, it creates distension, activates stretch receptors, and triggers the myenteric plexus to induce constriction. When it constricts at multiple different points. That creates like this little sausage link kind of appearance. Well, now let's go and think about what the next thing is going to happen. Here's a food bolus, which are going to represent in brown. Here's another food bolus, which are just going to represent in pink. Watch how it mixes these different things together. It's pretty cool. Now, I want you to kind of imagine we cut this in half. This food bolus, whenever this contracts at this point here, it's going to contract here. It's going to contract here and here, and you get the point, right? If we contract here, it's going to push this food bolus up here and this part of the food bolus down here. If we kind of do the same thing here, it's going to push this food bolus here and this food bolus here. So look at where it goes. So now this piece is going to go up into the top. That's this portion right here. So now this portion, when it constricted, it produces that. This portion is going to go down into the bottom. That's that portion. Okay? This portion here, whenever it constricts here, it's going to push this portion up. That's that portion, right? It's pretty cool. And now this portion, whenever it contracts here, it's going to push this portion down. That's going to be this portion. And you see the point. What happens as a result of this is now we're going to mix the actual chyme with other points of chyme along the length of the small intestine. That really helps in aiding this mixing process, as well as, to some degree, we're going to be moving this chyme towards the large intestine. So it plays a small role in propulsion and a very significant role in mixing, which helps with chemical digestion and absorption. Remember that. Segmentation primarily for mixing, which helps with chemical digestion and absorption, and to a very minimal degree, propulsion. Okay? All right, that's segmentation. Again, let's move on to the next thing, which is going to be peristalsis. All right, so now what we're going to do, guys, we're going to talk about two things here which play a significant role in the propulsive activity of the small intestine. That's called the migrating motility complex, which is primarily a peristaltic action. And then we'll talk about how peristalsis, general like peristalsis, also helps move chyme along the small intestine. Now, big thing I want you to remember, though, before we move on, is that segmentation, peristalsis, all of these things are altered by the sympathetic nervous system, the parasympathetic nervous system, the degree of distension, irritants. So I want you to also remember, don't forget, same thing when we talk about peristalsis and migrating motility complex, I want you to remember that distension, okay, so stretch. I want you to remember that the sympathetic nervous system, the parasympathetic nervous system, and also irritants play a significant role in the actual segmentation process. Irritants, they can help to stimulate. Parasympathetic nervous system is primarily stimulatory for this segmentation. Sympathetic is inhibitory, and distension is generally stimulatory. So again, same thing, this is the same concept when it comes to segmentation and peristalsis. All right? So, how does this all work? The first thing I want to talk about is you guys remember peristalsis, right? We talked about this in the video on the esophagus and how this plays a role. We also talk about this in the enteric nervous system. We're not going to go into crazy detail. I just want you to get an overall idea. Peristalsis is going to be trying to generally propel the actual food from the beginning of the small intestine down to the large intestine. That's the goal of it, right? How does it work? Very briefly, we'll go over the mechanism. We get a food bolus here, right? Here's going to be our nice big food bolus, right? So here's our food bolus. This dude is ready. And what's going to happen here is this food bolus is going to lead to distension. All right, so this food bolus, right, it gets into this area of the small intestine, right? So let's say it comes out of the stomach, goes into the small intestine, and triggers distension, right? So once it enters into the small intestine, it's going to stretch the walls of the small intestine. Now, if you guys remember, when you stretch the walls of the small intestine, it activates specific receptors that are located within the submucosa. And these are called your stretch receptors. These stretch receptors, what do? Well, they send signals to your myenteric nerve plexus. And if you guys remember, the myenteric nerve plexus, or Arbox plexus, is going to be in between the circular layer of the muscularis externa and the outer longitudinal layer of the muscularis externa. And these are both made of smooth muscle, right? So it's going to send signals outward here, and it's going to regulate the activity of this myenteric plexus. How does it do that? Well, if you guys remember, very, very simply here, we want the circular layer to cause contraction on the oral side of this bolus, right? So we're going to want to stimulate this portion here. So what's going to happen is we're going to stimulate this guy here. We're going to stimulate him, and he's going to allow for his axons to come over here and stimulate this portion. Okay, stimulate this portion of the circular smooth muscle. How does it do that? It releases chemicals like acetylcholine and substance P, right? That stimulates this circular smooth muscle layer to cause a constriction ring around that bolus on the oral side, okay? It's also going to inhibit this actual neurons that are going to control the longitudinal layer. Now, the longitudinal layer, whenever it contracts, it opens up the lumen. So we don't want the lumen to open up on this side. We want to close it off so that we push the bolus forward. So we're going to inhibit this and release things like VIP and nitric oxide. This side will now relax. Okay? If he relaxes, he's not going to open up the lumen, and he's going to keep that area tight, and it's going to allow for the bolus to move forward. But now, another thing that we have happen here is this: whenever we stimulate this circular smooth muscle right above this bolus, we need to relax the circular smooth muscle below this bolus. So how do we do that? Well, then it's going to send signals down this descending plexus. Okay? And this descending plexus here is going to release chemicals, right? Specific chemicals like VIP and nitric oxide, and that's going to inhibit this circular smooth muscle layer. When this circular smooth muscle layer is inhibited, what happens to the constriction ring? It doesn't develop, so it's going to relax. On the same situation here, you're also going to have the release of substance P and acetylcholine on the longitudinal layer below this bolus. And that's going to allow for this lumen to open up. So the whole thing that happens here is it allows for the bolus to move towards the anal side. Okay? That's the whole goal. So here's what I want you to remember to sum it all up. You got stretch. It stimulates the myenteric plexus. Above this bolus, in this case, towards the oral side, you want the circular layer to contract, longitudinal layer to relax. On the anal side, or below this bolus in this case, you want the circular smooth muscle layer to relax, and you want the longitudinal muscle layer to contract. In order to stimulate contraction, you have to release things like acetylcholine and substance P. In order to cause relaxation, you release chemicals like VIP and nitric oxide. That is the whole point of this peristaltic mechanism. The next thing I want you to remember here is that peristalsis, right? We said it can be affected by distension. It can be affected by the sympathetic, the parasympathetic nervous system, different irritants, even hormones. Here's what I want you to remember. Okay, so when it comes to peristalsis, right? The big thing you guys need to remember is that this is primarily a propulsive action. Okay, it's a propulsive action, and it basically what that means is it moves things from the oral side to the anal side. Okay, again, what things help to stimulate it? The parasympathetic nervous system enhances it, Distension can enhance it. Irritants can enhance it. Maybe certain hormones, which we can talk about later, can also enhance it. And the things that are going to inhibit it are primarily the sympathetic nervous system, right? Now, another type of peristaltic mechanism that I want to briefly mention, we talked about it in the GI motility, the stomach motility, and this is called the migrating motility or motor complex. Now, this is actually going to be regulated in specific times. Now, here's what I want you to remember. Peristalsis occurs whenever there's already food in the small intestine. The migrating motility complex, and this is super key, occurs between meals. So occurs between meals. In other words, it's a fancy way of saying that the person is in the fasting state. So that's kind of like a biochemistry term, right? They're in a fasting state. They're not eating. What the migrating motility complex does, and I want you to remember this, it's a peristaltic wave. It is peristalsis. It's a type of peristalsis, and it's a peristalsis that only happens between meals. This is occurring whenever food is in the small intestine. And what happens is it starts at the stomach, and it goes all the way to the ileum. Okay? Now, this process takes about 2 hours. It takes about 2 hours to get just from like the beginning of the small intestine, like the duodenum or the pylorus of the stomach, to get all the way down to the ileum takes approximately 2 hours. After it does that, it goes back and does it again. What's the whole purpose of this peristaltic process? When it contracts, it pulls with it any food residue. It pulls with it dead cells. So any of the cells that are basically damaged from the chyme or the acidic residue. It brings with it bacteria or any undigested like substances. For example, you're a little kid, they eat like a penny or something like that. The migrating motility complex is going to allow for that to continue to get moved along. Okay, so again, it's kind of a housekeeping function. Peristaltic process, can't stress this enough, occurs between meals in the fasting state. Starts at the duodenum, particularly. We say that it can occur at the level of like the pylorus of the stomach, body, pylorus of the stomach. But either way, from the point of the stomach to the ileum, it takes about 2 hours. It occurs about every 90 to 120 minutes. Okay? And it just continues to keep occurring. What does it bring with it? Food residue, dead cells, bacteria, undigested substances. It's moving it into the large intestine so that you can eliminate it. Okay? Last thing you got to remember is because this occurs during the fasting state, there's something different that stimulates it, not distension, not irritants. It can be, there's certain research that shows that acetylcholine can affect it. Don't worry about that right now. I want you to remember a particular hormone called motilin. It's released from your small intestine, and it's what drives the migrating motility complex. We don't know exactly what stimulates it. We just know it happens during the fasting state. And certain research has shown that acetylcholine works. The only reason they know that is because they gave people atropine, and atropine stopped this process from occurring, which is a blocker of acetylcholine. But big thing I want you to remember, motilin stimulates the migrating motility complex. What is the migrating motility complex? It's a peristaltic process that occurs after you've eaten a meal, between meals, moves from the stomach all the way to the duodenum, occurs every 90 to 120 minutes, and then repeats, brings with it food residue, undigested residue, and bacteria to prevent overgrowth. Peristalsis, general peristalsis, occurs whenever we've eaten because it's dependent upon distension. Okay? Segmentation also occurs when there's food in that area. Okay? So it's also going to be right after we've eaten a meal. All right, let's move on to the next and last part for the intestinal small motility. All right, so far we've talked about segmentation, we talked about peristalsis, we talked about particularly the migrating motility complex is another big one. Again, what I want you to remember, segmentation is primarily for mixing, not for propulsion. It does play a little bit of a role, but primarily mixing. Peristalsis and migrating motility complex are primarily propulsive actions. Big difference is that particularly migrating motility complex, is after, like, it's when you're fasting. It's in between meals. Peristalsis is actually going to be pretty much after you've eaten, okay? So while food is basically in that actual intestine. The next thing we need to talk about is how we get the actual fecal residue at this point in time, or pretty much you can say, you know, a watery type of fecal residue from the small intestine and the large intestine, because we have a muscle here, right? This muscle is called the ileocecal valve or sphincter. Let's actually write this sucker down here. So if we come down here, I want you to remember this muscle here. We have what's called the ileocecal valve, and really it's a sphincter. It's a circular layer of smooth muscle, okay? So I want you to remember that. Again, this is smooth muscle. It's very, very tightly constricted, smooth muscle. Now, one thing I want you to remember is that it's always closed. It's only open, okay? The only time it's open is under two circumstances, okay? One is due to elevated gastrin levels, okay? And we'll talk about what triggers that. And the other one is due to the gastroileal reflex, and we'll go over that as well. Basically, what I want you to remember about this is that these two things combined help with two overall goals, okay? One is to relax the ileocecal valve, and the second thing is to cause contraction of the distal ileum. And the goal of both of these things combined is going to lead to feces moving from the ileum into the large intestine. So let's take a look at the diagram and pretty much sum all that up. All right, here we go. Ileocecal valve, ileocecal sphincter. Let's pretend right now it's closed, and right at this portion, say right here at this distal ileum, is going to be some poo-poo ka-choo, okay? So some doo-doo right there. Now, what's going to happen is we need to move this from this portion of the ileum into this portion of the large intestine. What's this called? This is called the cecum. It's the first part of the large intestine. This little sucker hanging from the actual part of the cecum is called the appendix. Sometimes it's a little bugger that can cause appendicitis and cause a lot of problems for people. And then eventually you go upwards, right, into our ascending colon. So eventually we can say that the rest of it going upwards until we get to what's called the hepatic flexure is called the ascending colon. Now, we need to get things from the ileum into the cecum, past the sphincter. How do we do that? Well, let's come up to the stomach, believe it or not. Now, what happens is you eat, right? And you get this food now coming into the stomach. What is it going to do? It's going to initiate a stretch response, right? That stretch response is going to stimulate specific types of cells. Remember, you have those enteroendocrine G cells, and they start releasing gastrin. What does gastrin do? Well, it does a couple things. It can stimulate contraction, so it can stimulate the contraction of the stomach. It can stimulate hydrochloric acid production by the parietal cells. It even plays a little bit of a role in the pepsinogen secretion, which is eventually converted into pepsin by the chief cells. Okay, but another thing that it can also do is it can also regulate gastric emptying, so it can play a role in pumping the chyme, which is in the stomach, into the small intestine. But guess what else it does? It goes into your circulation, right? Travels through the blood because it is technically a hormone, and then it comes here to the ileum. And guess what it does to the ileum? It's going to initiate relaxation of the ileum. The ileocecal sphincter, relaxation of the ileocecal sphincter, but induce contraction or peristaltic activity. So it's going to lead to contraction of the distal ileum. So it's going to cause contraction of the distal ileum. And that's when it pinches here, what's it going to do? It's going to move this fecal matter from this portion of the ileum into the cecum as long as that valve is relaxed. Okay? Another mechanism, if you guys remember, you have these receptors, which are going to be different types of sensory receptors, right? And they can be stimulated by multiple different types of stimuli. It could be because you have acidic chyme. It could be because there's a lot of fatty chyme. It could be because there's excessive distension, right? A lot of these are different factors that might activate these sensory receptors. And once that sensory receptors are activated, it can send impulses along sensory fibers, primarily of like the vagus nerve. And this is going to come to your actual central nervous system, right? So this might be cranial nerve 10 if it's a long reflex arc, right? Which is your vagus. Activate specific fibers, right, of your efferent vagus nerve, which may come down here and do two things. One is it may induce relaxation of the ileocecal sphincter. And it may also induce contraction of the distal ileum. And what's that going to do? It's going to help to propel that fecal residue from the ileum into the cecum. What is this reflex arc called? We know it's a long reflex arc or a vagal, but it's actually called the gastroileal reflex. Okay? So I want you to remember that. Okay? So two things that are going to help with propelling this fecal residue into the cecum is going to be gastrin release and gastroileal reflex. Okay? That covers the motility of the small intestine.